pathogen? It is a disease-causing microorganism, and some of these could be, there are usually 4 common types, and these are viruses, bacteria, fungi, or protists. Okay, and we're going to go through those categories in the slides to come, and they depend on their host to provide suitable conditions and nutrients to grow and reproduce. So a host is basically the let's say the organism, because we know we could be talking about animals here, not just humans, the organism that is infected by the pathogen. So the host is, so we are hosts for pathogens because they infect us. We allow them to enter us and cause disease, and we provide them with suitable conditions and nutrients to grow and reproduce. Okay, and pathogens usually cause harm by producing toxins. Okay, so these harmful chemicals. So a toxin is a harmful chemical. Okay, so produces these harmful chemicals that damage cells and cause symptoms. Okay, these toxins can damage cells, human cells, animal cells, in various different ways. They produce different kinds of toxins which will have different actions. Okay, so they will have different actions. So not all toxins are the same. Okay, and the way that pathogens are spread are usually, contact with one another. So I don't know, if you come into contact, I don't know, kissing somebody and they're ill, that's how you could get it. They could be waterborne, okay, which basically means that they are transported in water, and therefore if we come into contact with any of that water that has a pathogen in it, we could get it. Or they're airborne. And what's the airborne one that we are currently at the hands of at the moment? COVID. So airborne, I'm sure you know what that means. It just means that it can be transported and spread through air droplets. Okay, so in case those words confused you, waterborne, airborne, now you know. Okay, and those are communicable diseases, because because again, they're spread, they are contagious and they can be passed on to one another. Okay, remember, non-communicable diseases are things that can't be transmitted between people and between species. Okay, so next slide. Viruses versus bacteria. Okay, so a virus, I don't know if any of you knew this, but viruses are technically classed as not being living things. Okay, they're a non-living particle containing genetic material. Okay, so why are they non-living? They're non-living, okay, because they don't do the main essential life processes. So they don't respire. Okay, they don't respire. They don't what was I going to say? So they don't respire. They don't really metabolize, which basically means they don't have lots of biochemical reactions taking place within them. So they don't do any of those, activities. They don't feed. Okay, they're not living. Okay, viruses contain genetic material in the form of something called RNA usually, and we'll go through what RNA is a bit later on. Okay, but you're not really expected to know what RNA is at GCSE. You'll do RNA when you get to A-level, definitely. Okay, but viruses, all they do is they contain this one material, which is usually RNA, which gives them the tools to survive. Okay, and the way that they do that is they have to depend on a host. Okay, they have to be able to get into a host and release this RNA in order to reproduce inside and cause all this damage. And they can't be killed with antibiotics. So we're going to go through the difference between antibiotics and antivirals at the very end. Okay, So that's why viruses would be classed as non-living, because they don't do the main life processes. So let me just write that there. Do not carry out main life processes. They contain this genetic material, okay? They just have a material inside them, this RNA, just a chemical. That's all it is, okay? Whereas bacteria are living organisms because they do all the life processes, okay? They feed, they grow, they respire, they metabolize independently, okay? Which basically means they can do that all in themselves and they don't need a host, okay? They don't need a host. To survive. So yeah, so that's why bacteria would be classed as living things, and these can be killed with antibiotics, okay? I'm sure everyone has taken antibiotics in their lifetime, but we've got to be very cautious now with antibiotics because of resistance, and again, we'll talk about that a bit later. And usually in bacteria, there's exponential growth, which leads to rapid infection. So basically it just means that they multiply at crazy rates, okay? Let's just write that down. Multiply at crazy rates. It's not writing brilliant today, this pen, actually, if it's connected all right. Yeah, okay. So that's what exponential growth means, and that will lead to rapid infection, okay? So let's move on. Okay, so again, that's just different kinds of bacteria and different kinds of viruses. You might get the flu virus, influenza virus, and you'll notice that they look very different, okay? So bacteria, they have all sorts of shapes. So Streptococcus that you can see here, it's all, come on. So it's all, they're all these chains of these circular globular things, okay? And E. coli, they're like rod shaped, they're long and like that. Whereas viruses, they usually have this coat on the outside. They have the material inside. This is the RNA, okay? And then they'll have all these spikes on the outside, okay? And that will allow them to stick to our host cells and get in, okay? So they look very different usually. So it's quite easy to tell the difference between a virus and a bacteria under the microscope. Right, viral disease. So you may have heard of measles, okay? You probably have been vaccinated against measles. Maybe if you were born in the 90s, it might be a different case because of the MMR vaccine, okay? Again, that's another scandal we'll talk about later. So measles causes the measles virus, and the symptoms of this is fever and a red rash, okay? So you can see on this person over here, they've got all these red spots, okay? And that's classic sign of measles, okay? And it's spread through the inhalation of droplets from sneezes and coughs. So it's airborne, okay? It travels in air and we can breathe that in, and that will give us the infection. And it's really serious. You know, it can be really serious and complications can be really fatal. So if you've not been vaccinated for MMR, then do it. Usually you are vaccinated during childhood. I think everyone basically is. So yeah, but we'll talk about actually how there was an outbreak of measles not long ago, and that's due to the whole MMR scandal, but again, we'll go for that a bit later. HIV. Now, I'm sure a lot of you have heard of HIV and know what HIV is and where you get it from. So if you didn't know, HIV stands for human immunodeficiency virus, okay? And it's a virus that attacks the immune system, okay? It attacks the immune system and it destroys our white blood cells, mainly T helper cells, okay? And we're going to go through the different components of the immune cells. T helper cells are one of them, okay? So we'll come on to that, but just take away from this that HIV, it attacks the immune system, it usually destroys white blood cells. And the way that, you know, HIV, you can get HIV and it's transmitted is as a STI, sexually transmitted infection. And usually that would involve body fluids, okay? So it could be blood, it could be you know, seminal fluid, any, you know, anything which involves sexual contact in some way. If someone is HIV positive and you have unprotected intercourse, then that could lead to HIV. And you get an initial flu-like illness. So because even though it's such a, you know, such a devastating illness, it's quite mild how it appears initially. It just appears as like a flu-like thing. So people might not think, oh, I have, you know, I have HIV. They might not have any reason to suspect they do because they've just got this very mild illness. But the longer that they leave it without getting tested, then the more severe it can become. And if it becomes so long that they don't get checked and they don't have any intervention, then people with HIV can go on to develop something called AIDS. Now, AIDS stands for Acquired Immune Deficiency Syndrome. And this is basically just chronic, just a chronic breakdown in the immune system. They just have no protection whatsoever, okay? They can't protect against disease anymore. Their immune system just completely breaks down. So it requires a course of lifelong drugs. There's no cure, but there'll be, you know, medication for the rest of their life in order to, you know, try and give them some sort of protection in the way of immune cells. So there is no cure. There's control. It's controlled with antiretroviral drugs. So, you know, to stop the HIV from replicating. Okay, so you know, the lower the viral load that you have. So viral load just means how much of the virus you have in the body. So it lowers viral load. Viral load, and that is how much of the virus you have in the body. Okay, so it will stop that, stops it from replicating, lowers your viral load, less likely to pass it on to another person. Okay. No vaccine, so there's no vaccine for HIV, but you know, there are you know, some miracle treatments that are coming through. So you might have seen on the news that there have actually been a few cases of patients that have been cured from HIV. There's generally there's not a cure for everybody, okay, because you know, these are very expensive treatments. It hasn't been approved for routine use, which basically means we can't give it to every single person that has HIV because it's just very expensive, a very time-consuming process. So we've got to try and find ways that we can overcome that, so we can give it to more people with HIV and give them the best chance at surviving and being cured of the disease. Yeah. So there are cases where people have used gene editing to try and cure HIV, to try and boost those immune cells again, but you know, it's not available to everybody. And the thing with HIV is, you know, prevention is best, so it's always good to take precautions. If you think that, you know, you might come into contact with someone that might be HIV positive, you can get something called PrEP now, which is called, I think it stands for prophylaxis exposure therapy. Okay, so basically people can take PrEP, and that is a, you know, cocktail of drugs essentially, and it will just stop any viral cells from entering into the body, and so in case you do come into contact with someone with HIV, you have those drugs in your body already, which is going to just stop them from getting into your cells. So there are things like that, but there's no actual cure for HIV if you do get it. Yeah. Okay, let's move on to talk about other viruses now. So tobacco mosaic virus. So this is a plant virus. Okay, it's a widespread plant disease. I think it was one of the first viruses in plants to be discovered, and it's caused by this tobacco mosaic virus. It's mosaic. Okay, it's got a mosaic-like appearance, which is also due to this discoloration. So you can see this leaf over here. It's got this patterned appearance, this mosaic-like appearance, because of all the yellow bits. So that's why it's called tobacco mosaic virus. and it usually affects growth due to reduced photosynthesis. So again, I don't think we've really done anything on plants yet. That might come in the new year, or it might come when you're in year 11. I'm not sure, but if you've not done plants, so basically photosynthesis is their way of producing energy, so glucose, so they can carry out respiration. So photosynthesis, it takes place in the chloroplasts of plants, which are these cells that contains this green pigment called chlorophyll. Okay, so chlorophyll is the green pigment in plants responsible for photosynthesis. Okay, so what you can see is, because of this yellow discoloration, this yellow color, it's not that green color anymore, so that means that there's probably less chlorophyll. Okay, there's less chlorophyll, so that means that there's going to be less less energy for respiration. They'll have less glucose, and they won't be able to respire as effectively, so that's going to affect their growth. Okay, so bacterial disease now. So let me just have a sip of water. So you've probably heard of Salmonella, form of food poisoning. So Salmonella, it's bacterial, and it usually results in these symptoms: fever, abdominal cramps, vomiting, and diarrhea. Okay, it's not very pleasant at all, and it's foodborne. So it usually goes, it can arise when food is not prepared properly in hygienic situations, not cooked properly. You know, if not cooked properly, you can get Salmonella. Okay, so be very careful of eating raw meat. And this Salmonella, basically, it secretes a toxin which will cause all these symptoms: vomiting and diarrhea. Okay, which will just cause you to essentially lose water from your cells, and that results in all this horrible vomiting and diarrhea. Okay, and a way that we can stop ourselves from getting Salmonella is we vaccinate the poultry, so where the meat comes from. So so chickens, for example, will be vaccinated against Salmonella. Okay, because that's where it comes from. Okay, this bacteria is living in the meat, in these chickens. So if we vaccinate them and protect them, then we can minimize the risks to us and prevent ourselves from getting it. Okay, so that's what we can do for things like that. We're talking about loads of STIs today. So one is gonorrhea, which is really a not very nice one, and I think if I actually showed you a picture of gonorrhea, you'd be scarred for life and come to these sessions again. So this is usually, it's characterized by thick green discharge from the genitals, and you get pain and stinging on urination, so it's really not pleasant. Okay, and again, that's an STI, so you get it from having sexual intercourse, and it can result in these horrible things. So it's always important to to wear protection, and to get regular testing, if you're very sexually active and things like that. So it's really not pleasant. And gonorrhea is controlled by antibiotics. Okay, so we will give people antibiotics if they have gonorrhea, because it's an infection, bacterial infection, and that will kill and get rid of the gonorrhea. And again, contraceptives, condoms, and other things, implants that girls can have, this will all protect against things like STIs. Okay, so fungal disease. So fungi. So this is an example of a fungus. It's called rose black spot. Okay, and this is what happens when that fungus infects the plant. It results in these black spots and it results in the leaves just dying. Again, I imagine it's got something to do with, you know, chlorophyll, and just completely stopping photosynthesis, and that's going to affect their growth and survival, and they'll just die. Okay, and this fungus is spread through water or wind, so it's carried in, you know, water droplets, carried in the wind, and if it lands on the plants, then it can infect the leaves of the plant, and they'll cause them to die. And you know, usually, and especially in farming situations, farmers will always give their plants lots of, you know, pesticides or fungicides to remove, you know, to try and kill off these fungi from infecting the plants, and they'll remove any effective leaves so it doesn't spread to any other parts of the plant. Okay, so that's rose black spot. Okay, last category of pathogen, protist disease. So malaria, I'm sure you all know what malaria is and where, you know, which countries are high risk for malaria. So with malaria, you usually have a very high fever. And I remember when I came back from Kenya, I felt really ill, flu-like symptoms, had a very high temperature. Actually, it wasn't that high. Actually, I think that was the reason why I wasn't diagnosed with malaria. Yeah, so I didn't have a high temperature, but I just felt very, very ill, flu-like. And I remember the doctor telling me, if you had a high temperature, I would have said it's malaria, and I would have tested you for malaria, but because of that, it's not. So, you know, usually a very high temperature, a fever, recurrent fever, which means, you know, it keeps coming on, is a sign of malaria. And what the malaria does is it damages your blood and your liver. Okay, and again, it can be life-threatening. So people, you know, lots of people sadly have lost their lives in places, very tropical climates, to malaria. And again, it's one of those things which is, you know, there's no vaccine for malaria. We can take anti-malarials tablets to, you know, stop the disease infecting us, but there's no real, you know, cure or real immunity from this. And the malarial protist lifestyle includes the mosquito. Okay, it's an animal vector. So a vector is basically an organism that carries the disease. Okay, so a vector, it carries this disease, and it will infect another person, another organism, by landing on it and then somehow injecting it with the disease that it's carrying. Okay, and you know, a way that we can stop malaria is we can do breeding prevention programs, you know, try and kill them off as much as possible. If we get rid of, you know, mosquitoes, then they have no way of transporting the malarial disease. Okay, and you usually see people have mosquito nets, again, to just stop, you know, any chance of a mosquito landing on your skin and infecting you. So it's a really, you know, not very nice illness, but there's been great progress made in terms of tackling this and coming up with a vaccine. Okay, so this is a summary then of what we've done. So what health is and the different types of pathogens. Okay, so now I'm going to do a few exam questions with you. And again, I'm not using Microsoft quizzes, I'm just going to do the solutions myself. But in a few slides in the next section, I'll get you to interact and have a go at thinking about how you would answer the questions. Okay, so rose black spot is a disease of roses. What type of microorganism causes rose black spot? So again, this is just simple recall. It's just trying to remember examples of bacteria, fungi, protists, and viruses. So you have to be able to basically give examples of these different types of pathogens and the diseases that they cause. So again, it's just learning that. So let's say we've learnt that, it's easy to kind of rule out which ones aren't the right ones. So we know that it's not a virus the only virus that we've done is tobacco mosaic virus. Bacteria, I don't think it's bacteria. I mean, you know, when we did bacteria, we talked about what did we talk about? We talked about gonorrhea and we talked about salmonella. So it's not that. Now it's either protist or fungus. The only protist disease that we talked about was malaria. Okay, so it's not a protist. So the correct answer is a fungus. Okay, so that's the correct answer. So that's quite a nice simple one. Okay, you just got to try and remember the categories and just be able to give examples. Oh, actually, it's coming up sooner than I thought, Menti. Okay, so we're going to try this. So what I'll do is I'll read the question aloud to you first before we go on Menti. Okay, so the question says, a student tried to grow some bacteria in the laboratory. The diagram shows some of the apparatus used. So we have an inoculating loop, a petri dish containing agar, and a bottle containing the bacteria. So the bacteria are in there, okay? And the student's trying to grow some bacteria in the lab. And this explains the method that he used. So he removes the lid of the petri dish, then he removes the lid of the bottle containing the bacteria, and then he uses the inoculating loop to pick up the bacteria from the bottle, and then he spreads that loop with the bacteria over the agar dish using the inoculating loop. Then he puts the lid back on, and then he puts it in an incubator in order for the bacteria to grow. Okay, and it says at the very bottom, steps, it's probably not very clear, but it says steps 1 to 5 could you cause the sample of the bacteria on the petri dish to be contaminated. So the key word is contaminated here. So we have to try and find ways that we can use to prevent any sort of contamination arising. So you've got to think about where would these sources of, you know, contamination come from. So I think one person put airborne and things like that. So that's absolutely right. So things in the air, if it's an airborne, you know, pathogen that's going to contaminate your experiment, your substance, what could you do? Well, you could make sure that you put on the lids of these petri dishes and the bottles as quickly as possible to stop anything from getting inside. Okay, so make sure you put on lids of petri dish and bottles as soon as you have finished with them. Okay, so that will just minimize the contact that it has with the air and anything, you know, getting inside. What's another thing that we can do to make sure we don't have any contamination? So think about what you would do at home. You know, what do you do at home, or what you should be doing at home? You know, if your parents do the cooking and stuff. So when you're, you know, cooking and preparing food, you don't just do it on a dirty surface, do you? You would wipe the surface that you're preparing food on. If you do it on a board or whatever, so you'd wipe down the surface. So in a lab situation, what would you be on? You might be on a table or a bench. Okay, so you could wipe down or sterilize the bench or table. Okay, so we've done 2 now. We've got one more thing to come up with that we can use to prevent contamination. So what other things that we could do? So we could look in the method, okay, and we could see if there's anything in the method where you think contamination could come from. So we've already talked about lids of the petri dish and lids of the bottle, so we can't talk that back over again. But have we talked about the inoculating loop? So we have not talked about the inoculating loop, and the inoculating loop is obviously the thing that we use to scoop the bacteria up and put over the petri dish. So what could you do beforehand to make sure that that inoculating loop doesn't contain any bacteria on it already? So you've got to make sure that it's clean, don't you? You've got to make sure that it is sterile. So you could be like, you could say sterilize the inoculating loop. So you can sterilize the inoculating loop with something like with ethanol. Oops. So alcohol is usually used to sterilize and kill off any bacteria, any contaminants on the loop. So that's what we would use. So, okay, so we've done 3 things now. So let's see if we've got the right answer. So it says any 3 from, okay, so there's quite a lot of things that we can say. So what did we say? So we said, make sure you put on lids of Petri dish and bottles as soon as you finish with them. So we have basically done that. We've done this one, haven't we? Minimize exposure of agar, culture, lift and replace the lid as quickly as possible. So that's 1 mark. Okay. Next thing we said is we said sterilize the bench. So that's what we said. We said disinfect the bench here. Wipe down, sterilize the bench or table. 2nd mark. 3rd mark comes from, so they said sterilize, so they talked about sterilizing the dishes or the agar, but we talked about this one, didn't we? The inoculating loop, but more specifically, we said it down here, dip the loop into ethanol. So that would get you the mark as well, okay? Those are the 3 easiest things that I can think of saying, or, you know, you could have said sterilizing the Petri dish or the bottle, okay? So we would get 3 marks for that, okay? So, next question. Why did the student grow the bacteria at 25 degrees rather than at 40 degrees? Okay, so I don't think we've really done, you know, the effects of temperature and other conditions on experiments and on, you know, processes that take place inside the body, but I'll explain it to you. So why did the student grow bacteria at 25 degrees rather than at 40 degrees? So these are the options. So the bacteria grew more quickly, so the bacteria grew more slowly. Well, we wouldn't want that, would we? We wouldn't want our bacteria to grow slowly. We want our bacteria to grow as quickly as possible. So that would not be the case. So the bacteria grew more quickly. Well, if that was the case, if we wanted, if the temperature was set at 25, so our bacteria could grow more quickly, then we would have it at 40 degrees, because obviously 40 degrees, so the higher the temperature, quicker the growth, okay, because it's going to speed up all those enzymes, those reactions that take place in your body. Higher temperature, more quicker reactions, more growth. So that can't be the case, because if the bacteria were growing more quickly, then we would have used 40, so it's not going to be that one, okay. To save money, that's just silly, because it doesn't mention anything about money in the question, okay. So anything that's financial, any financial reasons that they give you, just that's got to be wrong. So that just leaves us with this one. So to prevent the growth of a harmful pathogen, and that's exactly what's right. So I've basically told you, so the higher the temperature, the quicker the growth, the faster the reactions that take place inside, okay. So if you've got a higher temperature, it means you're giving more of a chance for bacteria to grow, okay? Because different bacteria grow better in different conditions. So if it's as high as possible, it's gonna allow for as many pathogens as possible to come in and grow. So that's why we've done it lower, so we can just make sure that only one bacteria will grow, one type of bacteria will grow. Okay, ensures one type of bacteria will grow, okay? So a higher temperature means you give more of a chance to other pathogens to grow, okay? So that would be the correct answer. Right, so this is the last question. So, hang on. Right, microorganisms can cause disease. Draw one line from each disease to the correct description. Okay, so again, this is quite a nice, easy one. How many marks is this? 3 marks, and it says that we need to draw one line from each, just one line. If you put 2 lines, then that's it. You're going to lose the mark. Okay, so you must make sure you read the question carefully. It's one line from each. Okay, so let's start with HIV. Okay, so we can just do it by process of elimination. can be spread by not washing hands thoroughly. I don't think that's right for HIV, is it? Because we've talked about HIV being spread through sexual contact, so it's not true. Can increase the chance of infection, such as pneumonia. Now, we haven't really discussed the different kinds of infections that you're more at risk of getting if you've got HIV, but you should be able to use your knowledge of what HIV does to the body and the immune system to work out the answer to this question. Okay, so I'll come back to that one on HIV. So malaria, let's just do malaria. So, okay, again, malaria not spread by washing hands. We know that it's spread through an insect. And malaria doesn't have anything to do with increasing the chance of infection, such as pneumonia. Part of the life cycle includes an insect. That rings a bell now, doesn't it? Because we said malaria is spread through mosquitoes. Mosquitoes act as vectors, so that is correct. Part of the life cycle of the disease includes an insect. Absolutely right. So the life cycle is obviously that malarial protist will reproduce, and then it's taken up by the mosquito. So that's the correct answer for that one. Salmonella, right? Salmonella is that foodborne bacterial disease that you can get. So treated with stem cells. We've not come across stem cells, so that's not the right one. Treated with fungicides, can't be fungicides because it's not a fungus, it's a bacteria. Spread by cough and sneezes, not true. Not true. Remember, it's foodborne. Okay, so we have to eat it, ingest it. So, but we do know that Salmonella can live on surfaces that are not wiped down properly, from where you've placed your uncooked food or anything. Okay, so it can be spread by not washing your hands thoroughly. Okay, so let's say you touch a surface that's had raw meat on, and after you touch that surface, you don't wash your hands. Then you use your hands to eat, or use it to touch other people, and then they use it to touch their face and eat. That's how you can get it. Okay, so that's right. So that just leaves HIV. So we've already done 2. Okay, HIV again, treated with stem cells. We've not discussed stem cells as so far at all, so that can't be the answer. HIV is a virus, so it's not that one. Spread by cough and sneezes. No, we know that it's spread by sexual contact, so that just leaves it with this. So HIV can increase the chance of infection, such as pneumonia, and that is true. HIV, you're at risk of getting a whole range of infections because you don't have an immune system. Okay, your immune system has completely stopped working. It's not functional. So of course that is going to increase the chance of any infections, such as pneumonia. Okay, so those are the correct answers. Okay, so HIV increases the chance of infection. Malaria, part of the life cycle, includes an insect, and Salmonella can be spread by not washing hands thoroughly.